Assalamualaikum everyone, my name is Cempedak. My student ID is 2024107213. I am presenting my final year project proposal for the Bachelor of Science Honours in Industrial Hygiene, Technology and Safety at the Faculty of Applied Science, University of Semarang. Today, I want to share with you a research project that addresses two pressing environmental challenges facing Malaysia: agriculture waste management and water pollution. My study title is Phosphoric Acid Assisted Thermal Activation of Cempedak, or well known as Artocarpus integer, leaf biochar for methylene blue removal and human exposure assessment. Let me walk you through the research, starting with the problem. Imagine this scenario. In Malaysia right now, farmers across 7,585 hectares of Cempedak farms are burning crop leaf as waste. At the same time, textile factories are discharging coloured wastewater into our rivers. We are facing two environmental crises at once. So, allow me to break this down. Agriculture waste, Cempedak or Artocarpus integer, is widely cultivated in states like Perak and Pahang, covering about 7,595 hectares of farmland. The shiny appearance on the surface of Cempedak leaf comes from a protective cuticular wax layer, which is mainly made of very long-chain fatty acids and their derivative. These waxy coatings make the leaf more resistant to microbial attack and slow down natural decomposition. So, the leaf litter tends to persist on the ground for a very long time. Because of this slow biodegradation, farmers often choose open burning as the fastest way to clear accumulated leaf, which releases smoke and particulate matter into the air and worsens local air pollution. Meanwhile, the textile industry discharges wastewater containing methylene blue, a synthetic dye that is highly toxic to aquatic life. Fish, plants and even humans can be harmed. Methylene blue is chemically stable and resists degradation, making it very difficult to remove using conventional water treatment methods. Traditional dye removal technologies like coagulation, advanced oxidation or membrane filtration are very expensive. Small industry and rural communities cannot afford them. So, what if you could solve all these problems at once? That is why I did my research. So, why hasn't anyone done this before? Let me explain the research gap. First, there is no prior studies on Cempedak leaf. Past researchers have successfully used fruit peels like banana peels, dragon fruit peels to make activated carbon for their removal. But Cempedak leaf have never been studied for this purpose. This is a completely novel application. Secondly, Cempedak leaf have natural advantages. Based on my findings, Cempedak leaf contains high levels of phenolic compounds and flavonoids. These compounds have negatively charged dyes, or we call this as anionic dyes, that naturally attract ionic dyes like methylene blue. So, chemically, Cempedak leaf are an ideal candidate to remove methylene blue. Thirdly, missing emission monitoring. While biochar production is promoted as environmentally friendly, the thermal treatment process releases gas and particulates into the air. No past finding has measured this emission during converting agriculture waste into biochar. As a student of industrial hygiene, I believe this is critical. We must ensure our solution doesn't create a new air quality problem. So my research filled these three gaps by studying Cempedak leaf activated carbon, optimizing the process and measuring air emission throughout. To address these gaps, I have three main research objectives. First, we need to synthesize effective Cempedak leaf activated carbon using phosphoric acid activation. I will use a chemical called phosphoric acid to treat the Cempedak leaf, then heat them to 500 degrees Celsius to create a porous carbon-rich material with high surface area for absorption. Secondly, we will evaluate how well this material removes methylene blue under different conditions. I will test how pH, adsorbent dose, temperature and contact times affect removal efficiency. For example, does it work better in acidic or alkaline water? How much adsorbent do we need? These are practical questions. Objective 3, we are going to measure airborne emission during the preparation process. Using personal air sampling equipment, I will monitor particulate matter and phosphoric acid mist released during the experiment. So now, how can I achieve this? My methodology has 5 main components. Firstly, we are going to synthesize the CLAC. I will collect fresh Cempedak leaf from local farm, wash them and dry them. And then I will soak the dried leaf in phosphoric acid solution using a 1:5 ratio by weight for 24 hours. This allows the acid to penetrate the leaf tissue. And then I will drain and wash the leaf, heat them in a furnace to 500 degrees Celsius under controlled oxygen condition for 1 hour. This creates the porous biochar structure. Finally, I wash it with acid and water to remove residual phosphate, then try it completely. Secondly, I will do batch adsorption experiment to test the effectiveness of the CLSC. Once I have the activated carbon, I will conduct pH experiments. I will prepare methylene blue solution at different concentration and pH levels, add my CLSC material, shake the mixture at conscious temperature for set times, then measure how much dye remains using UV spectrophotometry. I will use still oven or one variable at a time method. This means I change one factor at a time like pH or temperature while keeping the other constant. This approach is practical and transparent for student level research. Thirdly, throughout both the impregnation and termitification stages, a personal sampler will be worn by the experimenter to measure the amount of particulate matter emitted during the experiment. The filters will be weighed before and after sampling to quantify the airborne concentration, and this data will indicate whether the process complies with Malaysian occupational safety standards. And then, to fully understand the material I create, I will characterize it truthfully through 4 different ways, which is: first, I will use PET analysis, nitrogen phase option to measure the surface area and pore structure. Height surface area is critical for absorption. I will use SEM-EDX, scanning electron microscopy, to see the surface morphology and confirm the elemental composition. And then I will use FTIR, Fourier transform infrared spectroscopy, to identify the chemical functional group on the surface, particularly COP and hydroxy bond that form during phosphoric acid activation. And then I will use SRD, X-ray diffraction, to examine the crystalline and graphitic structure of the biochar. So, once I receive the adsorption data from the experiment that I have done, I will feed the kinetics to pseudo-second order models and the equilibrium data to Lamoille and Freundlich isotopes. I will use statistical test one way, ANOVA, to confirm that differences between conditions are significant at the P less than 0.05 level. So, what do I expect to achieve? I expect Cempedak leaf biochar to achieve greater than 95% removal of methylene blue from solution. This will rival commercial activated carbon performance. Next, I expect to identify that the best removal occurs at pH 8 to 10 mildly alkaline with an adsorbent dosage of 3 to 5 gram per liter and temperature around 35 degree to 15 degrees Celsius. This condition can be easily controlled in a treatment facility. Next, I expect baseline emission data. I will be the first to generate the first ever data on airborne emission from phosphoric acid assisted Cempedak leaf processing in a Malaysian laboratory settings. This supports compliance with occupational health standards. And then, if successful, this research will offer a low-cost adsorbent option for rural areas and small-scale textile factories that cannot afford expensive commercial treatment system. A Cempedak farmer or a small business could potentially produce their own adsorbent. By converting Cempedak leaf into an activated carbon adsorbent, I aim to demonstrate a practical, affordable and environmentally responsible solution to decontaminated water. Equally important, by monitoring emission throughout the process, I ensure that our solution does not create new environmental or health risks. This research is timely, original, and has real potential to benefit Malaysia's rural communities, for industry, and our environment. Thank you for your attention. This is the end of my presentation.